Welcome to this short video, which is a review of cell structures. The first observation of cells was conducted by Robert Hooke, and this was done on dried cork. And using a rudimental microscope, he was able to observe little compartments. And because it looked like monk cells, he described them like that. So he was the first to give the name cells. And a bit later, a cell theory was developed from many observations from many different researchers and with the development of microscopy. So in the end, in the middle of the 19th century, this theory was established and in fact named. And it says, in fact, it is in 3 tenets. So it says that all living organisms are made up of cells. The second tenet says that the cell is the fundamental unit of life. And the third tenet says that cells come from pre-existing cells. So the main people who established the cell theory are Leeuwenhoek, then Schleiden, Schwann, and Virchow, and this occurred because of the development of microscopy and because of their combined observations. So if we have a closer look on cells, first of all, it is very easy to tell apart a cell from non-cellular material because the cell is surrounded by a plasma membrane. So the plasma membrane is a double leaflet of phospholipids, and this double layer of phospholipids also contains a lot of proteins inserted and attached to both the external and the internal side. It is relatively easy to tell apart the internal side or the intracellular leaflet from the external leaflet because of the presence of specific components. So if you look at the cytoplasm, so the interior of the cell, and versus the extracellular fluid, so which is the watery environment that surrounds every cell, you will, at the level of the membrane, distinguish the presence of different molecular components. And the most important one is the fact that you have glycoproteins in the external leaflet, while you have filaments of the cytoskeleton, which give the architecture, on the internal leaflet and in the interior of the cell. If you also look between the phospholipids, so which are the grayish little molecules, you will see from time to time molecules based on the steroid structure, and these molecules are cholesterol molecules, and they are spread and scattered through the 2 leaflets of the membrane, and they have an influence on the permeability and the fluidity of the membrane. Now, if we look at the various types of cells that have been described and that are present on Earth, you have 2 big classes. One class is called the prokaryotes, and the other class is called the eukaryote. The main difference, or the most visible difference, we could say, is their size. Prokaryotic cells are extremely small compared to eukaryotic cells, and the second main difference is that the eukaryotic cells have internal compartments that are separated from the rest by the presence of membranes. So they have what we call membrane-bound compartments. In contrast, prokaryotic cells don't have this. If we look closer at the differences and similarities of these 2 types of cells, so first of all, the prokaryotes, they include the bacteria and archaea. They have a very small size. They are always unicellular, so they are never making tissues or working together. They can do colonies, but each individual of the colony is totally autonomous and doesn't rely on the others. And there are some differences in the chemical composition of membranes compared to the eukaryotes. We will see that in more specific chapters. Their genomic organization is simpler. They have no membrane enclosed compartments, I already said that. They have no nucleus. They have DNA. It is made, in fact, it is organized in a single circular chromosome, and this single circular chromosome is kind of folded and twisted and located in a region that is called the nucleoid, but this region is not embedded in a membrane. It is free floating in the cell. They can also have extra DNA pieces that are called plasmids and pili that are structure specific for some sort of sexual reproduction. On the other hand, eukaryotic cells include many cell types, so namely the fungi, yeasts that are also unicellular, protists, plant and animal cells that are usually making multicellular tissues, and they have a variable size. However, they are always larger than prokaryotes. They can be, as was said, unicellular or multicellular, but if we talk of plants and animal cells, for the most part they are multicellular. They have a complex genomic organization. They have membrane enclosed compartments. We call them in general organelles. So it is the general name for all the membrane enclosed compartments, so including the nucleus. And they also have filaments that direct the architecture of the cell, and this is called the cytoskeleton. If we have a closer look on prokaryotes, it has, as we said, a very simple structure. However, it is a very efficient structure because of its simplicity. It allows the prokaryotic cell to reproduce extremely quickly. It doesn't have a lot of structures and a lot of big complex molecules to reproduce, so it allows for a very quick cell division. It has a high diversity. If you look at the various types of bacteria and archaea that are present on Earth, there are many different types. They have different shapes, as you can see in the A, B, and C examples, but they all have the same organization, which is with the nucleoid or the DNA region, an inner plasma membrane, a cell wall, and an outer membrane that serves as a kind of protective layer. And again, the prokaryotes include archaea and bacteria, but no yeast, fungi, or protists, so pay attention to that. Prokaryotes are only archaea and bacteria. So let's now have a look on the structure of the eukaryotic cells, and we will compare animal and plant cells in this in the next few slides. So if we look at animal cells, we can recognize typical structures. So we will review them one by one a little after, but if we list them, you will find first the nucleus, then the endoplasmic reticulum, both the smooth and the rough endoplasmic reticulum, the Golgi apparatus, mitochondria, the cytoskeleton, and lysosomes and endosomes. If you look at a vegetal cell, you will find all the same things. However, you will, in addition, find a cell wall that is not present in animal cell. And this cell wall is composed of polysaccharides, or also called fibers. It will also have a big vacuole that is filled up with water. So it is a vacuole that will be responsible for the pressure, the internal pressure of the cell. And the last specific thing for vegetal cell is the chloroplast that is responsible for the capture of sunlight energy and responsible for transforming it into a usable form of energy for the cell. So all the structures that we just described each have a specific function and sometimes more than one function. Of course, these two cell types that we just described are very generic, but a cell that has specific functions would, of course, have more of the organelles that are needed to achieve the named functions. So for example, a cell that needs to produce a lot of energy will have more mitochondria. A cell that is responsible for a large amount of endocytosis would have a lot of endosomes and lysosomes. So remember that each cell type, depending on their functions, will have the number and the types of organelles adapted to the function the cell has to achieve. So we will first have a look on the nucleus and the nuclear envelope. As probably everybody knows, the nucleus is the place in the cell where DNA is stored. DNA consists in all the genetic information necessary for the cell to be able to perform its function and to sustain life. So, of course, because it is very important, it is kept in a confined and well-protected environment. So the nuclear envelope is exactly that. It has a double membrane, so it's not only a double layer of lipids, it is in fact 4 layers. You have 2 membranes enclosing the DNA. And this nuclear envelope is itself perforated with little openings that are called nuclear pores, and this will allow little proteins, ions, and molecules like RNA to be able to leave the nucleus and go into the cell in the cytoplasm, or in contrast, things from the cytoplasm can themselves enter in the nucleus, like for example a protein that would be necessary to transcribe DNA would be able to get into the nucleus by the means of the nuclear pore. The next step is the endomembrane network, which is in fact a network made of continuous and interconnected layers of membranes. In fact, if you look at the first one, which is called the endoplasmic reticulum, it is represented in blue, you will see that it directly starts from the membrane or the external membrane of the nuclear envelope. And if you look closer, you see that it has 2 different types. One type is called the rough endoplasmic reticulum. It has ribosomes attached to it. If you look on the right, the little yellow structure is a ribosome. It is attached to the endoplasmic reticulum, and because of its appearance, like with a microscope, you can see it like here. So it has little black dots everywhere at the surface of the membrane, and this is because of this rough appearance that we call it the rough endoplasmic reticulum. This is mainly responsible for the synthesis of proteins, so ribosomes attached to it, and they synthesize the proteins into the lumen of the rough endoplasmic reticulum. In contrast, if you look at the other part of the endoplasmic reticulum, which is also represented in blue, but you see that the stacks are smaller, so it looks more like tubes instead of large stacks. And this is called the smooth endoplasmic reticulum, because if you look at it with a microscope, you won't see the black dots combined to the membranes. The function of the smooth endoplasmic reticulum is to firstly synthesize lipids, so it is one of the main functions, and it also has an important function as a storage for calcium. So most of the calcium of a cell is contained within the tubes of the smooth endoplasmic reticulum. Directly after the endoplasmic reticulum, you will find the Golgi apparatus, which is a slightly different structure. It is again made of successive layers of membrane. They make like big flat bags, and they are oriented. You will see that there is a part facing the endoplasmic reticulum, which is called the cis Golgi network, and a face that is more oriented towards the plasma membrane, and this is called the trans Golgi network. and between the successive layers of cisternae, so this is the name we call the successive kind of bags of Golgi apparatus. So you will have vesicles that bud first from the endoplasmic reticulum and fuse with the cis-Golgi network, and then after, the vesicles bud again from one cisternae to the other, and they move up in the Golgi apparatus up to the trans-Golgi network, where they are distributed in the cell. So these vesicles will contain mostly proteins that are matured, folded, sugary additions are put on them, lipid additions can be put on them as well, and one of the very important function of the Golgi apparatus as well is to address the vesicles to the right place. So vesicles can be targeted to go outside, so it would be secretory vesicles, and they will contain a soluble protein that is released in the external environment. Or it could be membrane-bound proteins that will be put in vesicles as well, but the vesicles this time, when they fuse with the membrane, will leave the protein attached at the membrane level, and it will make a membrane protein. You can also have vesicles that go back to the nucleus or go back to other internal compartments if proteins are needed there. So the main function of the Golgi apparatus, in other words, is to mature, sort, and address proteins that are firstly synthesized in the rough endoplasmic reticulum, but then completely finished, if you want, in the Golgi apparatus. When they reach the top part, they are usually ready to go. The next compartment I wanted to talk about is called the lysosome, and the lysosome, as its name suggests, is a compartment where you will lyse or degrade stuff. Basically, when endocytosis occurs, a vesicle is internalized into the cell. It is the blue vesicle that you can see here on this picture. This endocytic vesicle will fuse with the lysosome, and you will also have vesicles coming from the Golgi apparatus, represented in red, that will as well fuse with the lysosome, represented in green, and the Golgi apparatus vesicle will provide proteins. Among these proteins, you will have mostly enzymes that will be here to cut, chop, degrade all the material that has been endocytosed. In addition, in the lysosome, there are proton pumps that will attract a lot of protons, and we all know that protons provide acidity, so it will keep an internal environment that is extremely acidic, so it will help with the degradation of macromolecules that have been internalized during the endocytic process. Let's talk now about the mitochondrion. The mitochondrion is another organelle that has very important functions. In fact, the main function we know for the mitochondrion is to provide energy. In fact, to take foodstuff or molecules coming from the food and convert them into a usable form of energy, which is ATP. So the mitochondria have a double membrane, a bit like the nucleus. So the external or outer membrane is a membrane that is relatively porous, but the internal membrane or inner membrane is a membrane that is much more tight, and it is extremely folded into cristae. So it is the name of these internal folds. And the internal environment is called the matrix, while the environment that is located between the two membranes is called the intermembrane space. So you can see here the intermembrane space, while the cristae here, they divide the internal matrix. Mitochondria don't only provide energy, they also have two very important functions. One of them is the synthesis, or at least in part, of steroid hormones. And the second very important function of the mitochondrion is to direct, or in fact control, apoptosis or cell death. We will see that in next chapters. The last organelle I wanted to cover with you is the chloroplast. It is an organelle that is specific to cells that are able to do photosynthesis, so plant cells. Again, it is an organelle that is embedded in two membranes, so a double membrane, basically 4 layers of phospholipids. But in addition to that, you also have very important and very developed network of a third membrane. This is called the thylakoid membrane. So basically here in a chloroplast you have the thylakoid membrane internally, you have the inner membrane surrounding it, and the outer membrane surrounding the inner membrane. The thylakoid membrane here folds many times. It looks a bit like stalks of pieces of money, stalks of coins, and these thylakoids, they contain a green pigment that is called chlorophyll, and the pigment is able to capture the sunlight energy and transmit the energy contained in high energy electrons into the cell in order to eventually produce energy. This closes the little review about cell structures.